Well, thank you very much, Peter, for that introduction. And let me say what a huge pleasure it is to come back to the Karolinska Institute. The last time I was here, I think, was about 3 years ago for a meeting on systems biology and cancer. You know, I'm always asked to talk about subjects about which I have no right to claim any knowledge whatsoever. Interestingly enough, at that meeting, I think quite a lot of other people thought exactly the same. We had a very interesting meeting. What I'm going to do today is to address Schrödinger's question, which of course it was the question, what is life? Those lectures were given in 1943 at the Dublin Institute of Advanced Studies, and they were, of course, the lectures in which Schrödinger first made his famous prediction that the genetic material would be what he called a non-periodic crystal. I still, and I think many others do too, still think that's an extremely good description, of course, of what was eventually found in the DNA. He also, though, drew a comparison between, on the one hand, physics, and on the other hand, biology. And his idea in that book was that they were really very different, because physics deals with order emerging from disorder. You have the disorder of the random movements of the molecules of a gas, but you also have the order of the gas laws and of thermodynamics. By contrast, he argued, biology is about order from order, because whereas the order down at the low level, it is in that aperiodic crystal. And I think his idea gave rise, of course, later on, or helped to give rise to the interpretations that were made of the early results in molecular biology of DNA when the double helix was discovered and when people began to work out what the coding was between DNA and proteins, which was to lead to the idea that there was, in a sense, in the DNA a determinate program. So unlike physics, you would go from particular events in molecules to particular events in the global property of the organism, that is, the phenotype. Now, of course, it's a hard thing to say that perhaps Schrödinger was wrong, but I think he was. And I would like to substitute that, the view that I and many others now express about biology, and it's certainly expressed in the book, The Music of Life, which is that indeed physics deals with order from disorder, but biology also must most of the time do the same, because what do we find down at the level of the individual gene expressions? We find stochasticity. If I take the gene expression levels in the ventricular cells of one region of the heart and compare them to the expression levels of those genes in an adjacent region of the heart, I will find that they vary. Now, of course, they vary to some extent according to the anatomy of the heart, and that is both well known and is also important in the functioning of the heart, but they also vary stochastically, even between neighboring cells. Stochasticity, therefore, has to be a feature of biology as well as order. Of course, we have a little bit of a dilemma, which I'll refer to a little bit later on, which is that we know well that there are certain changes that occur down at the molecular level, particular mutations, which will produce or be the cause of a major change in function at the level of the phenotype. But what has emerged in the research that's been done over the last decades is that those cases are actually quite rare. I find that in my work on the heart, there are certain modifications, certain mutations of particular ion channels that can produce a catastrophic situation, predisposing towards arrhythmia. But most of the time, the heart works perfectly well, just as the pancreas does, just as the brain does, despite all the variations that occur at the lower levels. So why should a physiologist be concerned about the question, what is life? First of all, to be concerned with that, you have got to be concerned with the central theory of biology, or the central story of biology, because I think that what we call evolutionary theory is actually a story, it's a history, which of course is the central idea, without which any form of discussion of the question, what is life, can't make sense. Moreover, the dominant theory of evolution over most of the 20th century and into this century was, of course, the modern synthesis, sometimes called neo-Darwinism, and that, of course, was a gene-centered theory of evolution with its determinate idea of there being a program that leads to the development of the organism. But in fact, organisms are extremely good at immunizing themselves from variations in their genomes. We're going to look at a few examples of that in just a moment, and some of the examples are discussed in that article in the Royal Society's new journal, Interface Focus. Let's just give a little bit of background from my own work. As the introducer said, I was responsible for working on some of the early discoveries of ion channels in the heart, and that led me to put this model of rhythm in the heart together in 1960. Here's the electrical potential showing rhythm that emerges as a cell property, not a molecular property, from the interactions between the various ion channels. In those days, we actually thought there weren't many more channels than there are in nerve. A sodium channel, 2 potassium channels, and a background leak. We now know there are many more. And in fact, later on, if we go now through to about 25 years later, in work that I did with Dario Di Francesco, we found there were many, many more potassium channels than we thought initially. And of course, by that time, Harold Reuter had made his great discoveries of calcium channels and of sodium-calcium exchange. We were also responsible in the work with Dario Di Francesco at discovering a new non-specific cation channel, which is responsible for this phase of the rhythm, and also for clarifying the role of the sodium calcium exchange. Now, that's all technical detail, and I don't want to dwell on that. What I want to do is to use that model to perform a very interesting experiment. So what we're going to do is to use a version of that model that was developed for the rhythm generator of the natural rhythm generator of the heart, which is the sinus node, and I'm just plotting 2 of the channel properties, a background sodium channel and the non-specific cation channel that I referred to earlier. This one normally contributes about 80% of the depolarizing current. It is therefore the rhythm generator, if you like. And as you knock it down, you can notice 20%, 40%, all the way to complete knockout. You might expect a very large change in frequency. In fact, the change in frequency is very modest, only about 10%. And what is happening, of course, is the system is allowing this other mechanism to kick in and to keep the rhythm buffered, as it were, from the change at the lower level. And you can even use the physiological interpretation of what is going on to work out what is happening, because if you draw a line along those voltage traces, you can see that the traces at this end are moving into a more negative range of electrical potential, and it's precisely that that activates the other channel to kick in and to keep the rhythm fairly constant. And that is a general property of cardiac rhythm. It's a general property of other rhythm generators too, for example, of circadian rhythm. There are backup mechanisms. Many more examples of what you might call knockout that don't actually succeed in producing a very large phenotypic effect. This is from some work, again fairly technical detail, but I won't dwell on the technical detail other than to say that a targeted knockout done in experiments first and then by simulation reproducing the experiments of Ken Philipson, targeted knockout of the sodium-calcium exchange produces almost no change in overall electrical function. Now, is this a specific property of some of Denis Noble's work on the heart, or is it general? I was delighted, therefore, when I saw this systematic study by Hillenmeyer and his colleagues in yeast working through the 6,000 genes and producing what they called a chemical genomic portrait of yeast. What did they find? Genetic buffering is general. 80% of the knockouts are silent under normal physiological conditions. That doesn't mean to say that those genes don't have a function, far from it. They do code for a protein. That protein is produced. Of course, what you have to do if you want to reveal that function is to stress the organism, for example, with metabolic stress, and then you can begin to reveal the function. There are many more examples. I won't dwell on that other than to say that if you want to understand regulators in biological systems, which is part of an answer to the question, what is life? Life is a regulated process, then you don't get that most of the time from knockout experiments. As I said earlier on, sometimes you do. Biology, therefore, is a mix. Occasionally it is order from order, but often it is also order from disorder, in a sense just like physics, though I think there are some important differences as well. For those who want to follow up on that question, this lovely article in Bioessays is worth reading. So what is going on? And here I want to also digress a little bit on the concept of a gene, because I think two things are going on here. First of all, extensive buffering of organisms against genomic change. That's now documented, I think, sufficiently well to say that it's a fairly general phenomenon. But there's another major concept that I want to introduce, which is that the concept of a gene has changed, and changed in a very significant way indeed. And it's central to understanding what is wrong with current evolutionary biology theory, that is the neo-Darwinist theory. The way I explain this is to say that we have DNA, obviously, we have a phenotype. Of course, it's a bit artificial to draw complete distinctions between the two. As I will quote with Barbara McClintock later on, the DNA is also, after all, a part of the phenotype. But I think we all know what we're saying here. Here's the genetic material in the sense that the molecular biologists would use that term today. Here's the interaction with the environment. Here is, if you like, the complete phenotype. And what we know is, of course, that all of these interact in bidirectional ways. That's also important with the biological networks, the signaling pathways, the filters, the conditioners, and so on, which actually generate the function. If we want an answer to the question, therefore, what is life, it's inside here. If I take the DNA out of a cell, if I could do just that, and put it into a Petri dish, I could keep it for 10,000 years and it would do nothing. It's the rest of the system, of course, that tells the genome when it wants to make particular proteins. It's the rest of the system that gives the dynamism. Now I come to the point about the definition of genes. When Johansen in 1909 first introduced the word gene, of course following in the footsteps of Mendel, who didn't use that word, but he had exactly the concept, what he actually meant was a discrete inheritable phenotype. What we now mean, of course, by a gene is a particular stretch of DNA. Now, the important point I want to make is that not only are those two definitions not the same, that's obvious from the fact that this only includes this part of the diagram, whereas this includes the whole diagram, they are conceptually different too. Because Johansen's definition is all-inclusive. He actually says that in his article. He says it's a something. It doesn't really matter whether it was DNA or not. Of course, they did not know about DNA at that time. Incidentally, Richard Dawkins uses exactly the same definition most of the time as Johansen. He also says it is, as it were, a general concept. It is, if you like, a catch-all concept. But of course, the problem with catch-all concepts that say and include everything is that they can't be tested. It is a necessary truth, a conceptually necessary truth, that a gene defined in this way as the cause of a particular phenotype characteristic is just that. It is the cause. You don't ask the question, is it a cause? It is so by definition. Now, the situation, of course, with a definition of a gene that is done in terms of a DNA sequence is that precisely that question can be asked. There is an empirical question to be settled, which is, does that particular piece of DNA have a big effect on the phenotype? You can perform that experiment with the kinds of knockout experiments that I've already described. So the difference is fundamental. Now, where is the deep problem in the neo-Darwinist thesis, particularly as expressed by people like Richard Dawkins in popularizing it? The problem lies here, that if you want to distinguish clearly and unambiguously between the replicator, which is, of course, the DNA, and the vehicle, which, of course, is the organism that carries the DNA, then you must use this definition of the gene. If you use this one, the one that Johansen first introduced, then it doesn't make sense to draw a distinction between the replicator and the vehicle, because this definition includes much more than the DNA. And I don't think that point has been properly appreciated. In relation to knockout experiments, of course, to represent the fact that most knockouts and mutations are buffered, but knocking out just one gene may not get through to the phenotype because it gets absorbed, as I showed in those computations, but also in the experiments on which they're based a bit earlier. But 2, of course, may be enough to get through, or you may need to do 3. Who knows? That's a matter open to empirical investigation. So I want to turn now to a few other aspects in which the answer to the question, what is life, taking particularly evolutionary biology as absolutely central to that question and to answering it properly, poses today some very big problems. But what we need to do is to first go through a very brief history of evolutionary biology. And I start by going back just over 200 years to Jean-Baptiste Lamarck, who published the Zoologie philosophique, his great work in 1809, exactly 50 years, incidentally, before Darwin's Origin of Species. Jean-Baptiste Lamarck actually invented the term biology, biologie. He was one of those who first, as it were, therefore defined our subject as a separate subject from other natural science subjects. But he also did something else for which he is very famous. He certainly espoused and popularized and used the theory that there was inheritance of acquired characteristics. and many of you will know that, of course, it is a clear feature of the modern synthesis or of neo-Darwinism that that process is excluded for reasons I'll come on to a little bit later. Darwin, of course, published his Origin of Species in 1859, proposed, of course, the theory of natural selection. Of course, neither he nor Lamarck or anybody else at that time had a theory of genetics and heredity in that sense. They didn't know of Mendel's work until it was rediscovered later. Now, an important point to make. Darwin also assumed the presence and function of the inheritance of acquired characteristics. It's present in the Origin of Species in 12 different places, unambiguous, all documented, incidentally, by Ernst Mayr many years ago in his great book, Biological Thought, in 1982. Further, Charles Darwin, in I think it's the 4th edition of the Origin of Species, specifically praises Lamarck as this great biologist who championed the idea that there was transformation of species originating from a common ancestor. Darwin was not a neo-Darwinist. That's a very important message. We'll come on to the significance of that a bit later on. So what gave rise to neo-Darwinism? What gave rise to neo-Darwinism was, of course, the work of Weismann, who introduced the idea, which we now call the Weismann barrier, that is the specific exclusion of the inheritance of acquired characteristics. And the idea was, of course, that the inherited material, wherever it was, and of course he didn't know where it was at that time, is immune from the rest of the system. The rest of the system, however it develops in the lifetime of an individual, cannot influence the genome, as we would now call it. And of course that later became encapsulated in Francis Crick's idea of the central dogma of molecular biology. Let me just also make a brief explanation at this point. In the paper in which he defines the central dogma in 1970, Crick was actually very careful. He referred to the fact that, of course, DNA sequences code for protein amino acid sequences, but amino acid sequences do not code for DNA sequences. In that sense, there is a unidirectional flow of information. But he did not make the mistake, which was made later by Dawkins and many others, of assuming that that meant that no information passes in the other direction. And of course, if you ask the question, what makes your heart cell different from your pancreatic cell, different from your liver cell, it is precisely the passage of information from the system onto the genome to tell the genome to make a completely different pattern of gene expression in a heart cell, a pancreatic cell, a liver cell, a bone cell, and so on. So we know already, even just from that observation alone, that there must be information passing the other way. We'll come on to the significance of that also a little bit later. All of this was put together by Julian Huxley, Fisher, Haldane, Sewell Wright, and others to form what was called the modern synthesis, and it's the modern synthesis that is often also called neo-Darwinism. Now, let's look at the popularization of it in The Selfish Gene. I should say that I've interacted with Richard Dawkins on these issues now ever since he published The Selfish Gene. In fact, I've interacted with Richard Dawkins from even before he published The Selfish Gene. What is it? It is, of course, a gene-centered view of natural selection. That is what the modern synthesis is. Accumulation of random mutations. By random here is meant not related to function. Random compared to function, then followed by selection. That means, incidentally, that physiological science has nothing to say about the variation in genetic material. Very important, because we'll see that that view is actually wrong. Then the impossibility of the inheritance of acquired characteristics, a view, of course, inherited initially from Weissman and the Weissman barrier, and then later on buttressed and supported by the more popular interpretations of the central dogma of molecular biology. Now, I say that's miscalled Lamarckism because actually, if you look at the history of the idea of the acquisition of characteristics that can be inherited, that idea was not Lamarck's invention. Many others before Lamarck had said the same thing. Lamarck, like Darwin himself, just simply assumed it. They didn't invent it. Neither of them did. The other point that I've emphasized earlier on is the distinction between the replicator, the genes, and the vehicle, the phenotype. And notice what I said earlier on, I'll just repeat it now. The only way in which that distinction can be clear is if the gene is defined as DNA, as what we would now today call the replicator. So the vehicle is separate from the replicator. If, as Richard also does, you see, you define gene to be the original definition by Johansen and implied, of course, by Mendel, though he didn't use that word, then you cannot make that distinction. There's no way of having it both ways. You either define genes in terms of the DNA replicator, and then you have a clear distinction between replicator and vehicle, or you define genes in the way that Johansen originally did in terms of phenotype, and then you can't have such a clear distinction. And as I said earlier, these ideas were buttressed by what I would call misinterpretations, because I don't think they were intended by Francis Crick, misinterpretations, the central dogma of molecular biology. Now, the main point of my talk today on the question, what is life, is that to answer that question, we have to realize that all of these rules have now been broken in ways that I'll just go through. First of all, are mutations random? The answer is no, and by a very long way. In his book, recently published, the book by Jim Shapiro in Chicago, Evolution, a View from the 21st Century, writes, it's difficult, if not impossible, to find a genome change operator that is truly random in its action within the DNA of the cell where it works. But I want to go a bit further than Jim does. Jim documents with very, very many examples from the empirical literature, scientific literature, the non-random nature in the sense that there are hotspots in the genome, there are great big reorganizations of the genome, whole domains, as we will see in a moment, that have shifted around in the course of evolution. But I think he fails to make an important distinction which has to be added to this, which is the big question that should worry the neo-Darwinists is some of that non-randomness functional? In other words, can the functional state of the organism influence the DNA? And we'll see in a moment that it can. He also makes the point, which I want to emphasize, that if you look at what emerged from the first sequencing of the human genome, you find a very interesting fact. That for two major classes of proteins, transcription factors and chromatin binding proteins, the evidence is that they could not have evolved one mutation at a time in a gradual way, as is in accord with the spirit, at least, of the modern synthesis or neo-Darwinism. This diagram is a little bit technical, so I'll just go through it very, very quickly. All you need to know is that these are representations of particular proteins in different organisms, all the way from yeast through worm through to the human. And the stars that I'm going to put up now are whole domains, that is, long stretches of DNA that have clearly moved around in the course of evolution as whole domains, not a gradual accumulation of mutations, point mutations or small mutations. And as you can see, there are many of them. And the same is true for the other class of proteins that they studied. So proteins, not all of them anyway, did not evolve just via gradual mutation. Something is enabling the system to reorganize the genome. So the question I'm going to ask now is what happens in this view to selfish genes? And again, I come now to a deep conceptual error. The problem with the selfish gene is very simple. The book is written beautifully. Anybody who's read Richard Dawkins' books must admire the skill that he has in expressing the view that he expresses and the way in which he uses language. They are brilliant books. But when he was asked the question, is selfish, in the title of The Selfish Gene, metaphor by the philosopher Mary Midgley, he wrote back, "That was no metaphor. I believe it is the literal truth." But then he went on to write this very significant qualification. Provided certain key words are defined in the particular ways favoured by biologists, and if you've been thinking about the question, what is a metaphor? A metaphor is precisely taking a word from one context and changing it and making it perform a new function. It is a way, if you like, of redefining a word. Clearly, in writing this, he didn't understand the question that Mary Midgley was putting to him. Was it a metaphor? It clearly is. He didn't think it was. I wrote about this more recently in an article published in the Journal of Physiology just 3 years ago, and a quote from it is that a metaphor does not cease to be a metaphor simply because one defines a word to mean something other than its normal meaning. It indeed is the function of metaphor to do precisely this. And so, at the heart of the neo-Darwinist synthesis, at the heart of its popularization, there are a number of major conceptual mistakes. First of all, a mistake about what is the real definition of a gene, and it slips between the two according to what the question is, and you can't do that. You can't do that and be logical. And second, because, as I show in that article, but I don't have the time to go through all of that in the lecture, it's actually impossible to demonstrate any biological experiment that would distinguish the selfish gene theory from any alternative. And incidentally, curiously enough, Richard Dawkins agrees with that. In Extended Phenotype, he writes specifically, "I doubt whether there is any experiment to prove my case." So that aspect, at least, of the neo-Darwinist approach to the question, what is life, is simply not falsifiable. Now we come to the big question, and the difficult one: can acquired characteristics be inherited? I discussed this a number of years ago with John Maynard Smith. We were both at a Novartis Foundation symposium. He published a lovely book called Evolutionary Genetics in 1998, and he made a very interesting statement. He said, John Maynard Smith, although he was working totally within the neo-Darwinist view, he was, I think, a great thinker. And he said in that book specifically, you know, Lamarckism is not so obviously false as it's sometimes made out. Let's have a look. He was absolutely right, although he didn't know that at the time. There are many examples now. Those who want to go to my Music of Life website will find a page devoted to what are called answers to very common questions on this central issue of the theory of evolution and the question, what is life? And although I'm just going to give a few examples in this slide, there are many more on that page. Weaver's work looking at the way in which you can mark the genome by behavior in each generation, and that can work even if nothing goes down through the germline. Anway and his colleagues looking at endocrine disruptors where the process does go down through the germline, and we think probably through inherited RNAs. This paper also showing male transgenerational responses in the human. Beautiful paper by Yunhua Sun and his colleagues at the Fish Institute in Wuhan in China, doing cross-species cloning. I'll show that in a moment in another slide. And an extraordinary study by Rehavi and his colleagues in Cell, published around 3 years ago, showing not only transgenerational inheritance of an acquired RNA-based response, but also that it persists for 100 generations. They followed it in C. elegans for a whole year. The usual reply of neo-Darwinists to these examples is to say the effects die out. Well, they don't always die out. And then they will say, well, it's rare. Indeed it is, as far as we know at the moment. But remember also that speciation is rare. It doesn't have to happen very often. It hasn't happened in our lifetimes. It hasn't happened in the lifetime of humans. In fact, we've tried very hard with artificial selection of dogs, cats, fish, and so on. We've produced new varieties, but never new species. So, environmentally induced changes can be inherited. I think the evidence for that, as I said, is now growing very rapidly. Go to the answers page on the Music of Life website if you want more details. I said I'd say what happens in cross-species clones, and then we come towards the end of the lecture. What Jung Hassan and his colleagues did in Wuhan was to take a carp, which is this one, a goldfish, and proceeded to take the nucleus from a carp and insert it into the fertilized but then enucleated egg cell of the goldfish. You should get unambiguously a carp if the neo-Darwinist view is correct. What you get is intermediate. This is the result. It's one of the few examples where cross-species works in the sense that you produce an adult. And what it produces is an adult whose anatomy is intermediate between the two. You can even quantify that. If you look at the total number of vertebrae in the goldfish, it's about 26, varies up or down 1 or 2, and the carp is about 33. In the cross-species clone, you get a figure in between, but it's closer marginally to that of the goldfish. There are other respects also in which what they call the inheritance of cytoplasmic information is clearly occurring. And I want to come now to one of the greatest thinkers about genetics and their relation to the phenotype. I have a lovely picture in my office in Oxford of Barbara McClintock at the age of about 81, I think, receiving the Nobel Prize here in precisely this lecture theatre, standing exactly where I'm standing at the moment, and she was giving, of course, her Nobel Prize lecture, which she received in 1983. She was, of course, the person who discovered what we sometimes call jumping genes or mobile genetic elements, but she went much, much further than that until she was, I'm sorry to say, prevented from publishing. From 1953 onwards, she no longer published on this issue until, as far as I can recall, she was awarded the Nobel Prize in 1983. She wrote in that lecture, in the future, attention will undoubtedly be centered on the genome and with greater appreciation of its significance as a highly sensitive organ of the cell. The result of her work on maize was to show her just how labile the material is, the genetic material is, in response to environmental events. And she wrote, responding to those often by restructuring the genome. Here we come back to what Jim Shapiro was drawing attention to, that clearly during evolution there have been major restructurings of genomes, movements of whole domains corresponding to various proteins. So cytoplasmic changes can be inherited, and I think I'll jump straight to the next slide just to give one example of how strong that inheritance can be. This is the article I would recommend reading if you want what I would regard now as the best example of that, published by Joe Nadeau's lab, at that time in Seattle, in PNAS at the end of 2012. They write, I won't go through the full details, the heritable epigenetic changes persisted for multiple generations and were fully reversed after consecutive crosses through the alternative germline image. So my main conclusion, environmentally induced changes can be inherited. To the question, are they fairly rare, I would give the response I gave earlier on, so is speciation. So when we ask the question, do we know what the precise mechanisms of speciation were, I think the honest answer is that we don't know. Not yet anyway. Now I come to my final conclusion. If functional changes in the adult can be inherited and therefore a target for natural selection, then the analysis of function is highly relevant to evolution and, of course, highly relevant to defining the question or an answer to the question, what is life? Because, as I said earlier on, this area of biology is central to understanding the answer to that question. I think what I want to do is to leave time for discussion. I'll just jump the remaining slides. They just simply summarize what I've already just said. And the synthesis that I would propose is shown here, that we have to recognize that selection is multi-level, that acquired characteristics can be inherited. To quote Barbara McClintock, the genome is an organ of the cell, not its dictator, and that genomes are not isolated from the organism and environment. Those who want to listen to another version of this lecture, it's on the Music of Life website and on YouTube. And the article there, published just last year in Experimental Physiology, also gives chapter and verse on almost everything that I've presented today. And I finish with this lovely picture that I used right at the beginning. Some of you will recognize the left-hand picture as Conrad Waddington's beautiful picture from The Strategy of the Genes, in which he tried to explain the way in which there is plasticity at the developmental level in the phenotype, which is representing by a landscape here, as the consequence of various gene products interacting in networks that, as it were, are situated between the genome level and the phenotype level. Now, I know that's a bit artificial. We can argue about the exact definition of a phenotype and so on. But he's got the message. He's got exactly the system's approach to it. What happened to Conrad Waddington, he was largely ignored, pushed to one side, just as Barbara McClintock was until she received the Nobel Prize. So my final question is, who pulls the strings? In any multi-level set of interactions in any biological organism, the answer is everything can. If you take a three-dimensional network, just put together some knitting and make a three-dimensional knitting structure, you pull any bit of it, the whole network changes. All networks do that. So it doesn't make sense to isolate one part and say that is the cause. If you have networks as part of the definition and processes in those networks as part of the definition of an organism, then the answer to the question, what is life, is it lies in those processes in the networks. There is no privileged level of causation. And with that, thank you very much. The end of the lecture. Thank you.